Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Dr. Dileepa Jayawardena. Biofuel is an important plant-based resource to supplement or replace the use of fossil fuels. However, when land for farming and farming resources can be scarce, it can cause tension on which crops farmers should focus on. In this episode, Dileepa joins me to discuss his research growing biofuel crops on marginal lands, reducing that tension by moving them to areas not normally used for crop production. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, and welcome to the show. Today we have Dileepa Jayawardena with us. Dileepa is a research associate in the Great Lakes Bioenergy Research Center at Michigan State University. Before joining Michigan State University, he worked as a postdoctoral fellow in the Cropping Systems Agronomy program at the University of Idaho. He got both his biology master's and biology PhD in the ecology track from the Department of Environmental Sciences at the University of Toledo. Hello, Dileepa. How are you doing today?
I'm doing great, Abby.
Wonderful. Well, we're so happy to have you. So we are going to be talking about biofuel crops today. So to get us started, I was hoping you could kind of talk about biofuel crops in general, kind of how they work, what plants are used, things like that, as well as how that relates to the overall research questions that you were hoping to address.
Sure, Abby. Let's talk about the bioenergy crops. So in general, bioenergy crops can be divided into three main generations as first, second, and third generation bioenergy crops. The first generation bioenergy crops include the annuals such as corn and soybean. The second generation bioenergy crops include herbaceous perennials and woody perennials, and these are collectively called the lignocellulosic crops or simply cellulosic crops. And the third generation of biofuels actually mainly produced from algae. So when producing biofuels from the first generation bioenergy crops, for an example, if we take corn starch is used. So currently in the United States, about 15 billion gallons of ethanol is produced from the first generation bioenergy crops, and most of that, about 95% of that actually comes from corn starch. So corn starch is a carbohydrate, and it's made up of polysaccharides. And if we break down the polysaccharides, what we get is the building block of the polysaccharide, which is the sugar molecule. So if we ferment the sugar, what we get is ethanol. So that's how simply ethanol is produced from corn starch. The second most common biofuel produced from the first generation bioenergy crops is biodiesel. So currently in the United States, about 3 billion gallons of biodiesel is produced. And most of that biodiesel actually comes from soybean, over 90%. So during the production of biodiesel from soybean, we simply react the fatty acids found in the vegetable oil with an alcohol, and that generates a mixture of alkyl esters, and this is what we call the biodiesel. So this process is called the transesterification. So those two are the most common forms of biofuels produced from first generation bioenergy crops. In second generation bioenergy crops, when we produce biofuels from second generation bioenergy crops, we follow a couple of methods based on the temperature, namely high temperature deconstruction and low temperature deconstruction. And we are mainly interested in the low temperature deconstruction, which is done with the involvement of enzymes or chemicals. So if you recall your plant biology classes, you learned that the plant cells are actually surrounded by cell walls, and the cell walls are made up of three polymers: cellulose, hemicellulose, and lignin. So what we try to do when producing biofuels from second generation bioenergy crops, or also known as cellulosic bioenergy crops, is to convert that cellulose into the biofuel. So just like the polysaccharides found in starch, cellulose is also a polysaccharide which is made up of sugar molecules. So cellulose is specifically made up of glucose molecules, and hemicellulose is also a polysaccharide which can be used to produce biofuels because it's made up of 5 carbon sugars, mainly from xylose. So if we break down these polysaccharides, what we get is the sugar molecule, and then if we ferment that with a fermenting agent like yeast, or sometimes we can use bacteria such as Zymomonas species, we get what we get is ethanol. So that's how ethanol is produced from the second generation bioenergy crops. And the third, the biofuel production from the third generation of biofuel crops is similar to the transesterification process that I described when talking about the biodiesel production from the soybean oil. So that's how different biofuels are produced from different bioenergy crops. So the current global human population is about 8.1 billion, and by the year 2050, it's projected to be somewhere around 9.7 billion. So that means we need to increase the food production by about 50%. But on the flip side, the area of the arable lands available for food production keep declining, mainly due to intensive management practices as well as climate change. So on one hand, we need to increase the food production, but on the other hand, the arable lands that can support the production of food keep declining. So in order to get the maximum use out of the currently available arable lands to increase the food production, we have to we cannot dedicate any area of the land, arable lands, for biofuel production. So if we do so, then there will be a food versus fuel conflict. And also someone could argue that okay, if we are running out of arable lands, why don't we expand the agricultural lands by clearing the native habitat? So this is what we call the indirect land use change. So it has even worse consequences because these native habitats contain carbon stores, and if we clear them, they get exposed to the atmosphere and then oxidized and released back into the atmosphere as carbon dioxide. So we know that carbon dioxide is the main culprit behind the current global climate change, and if we want to recapture the released carbon dioxide, it may take even decades or even centuries. So that's why we need to come up with an alternative source, and that's why we believe cellulosic crops have the potential to produce biofuel sustainably, because biofuel production with the first generation bioenergy crops seems not sustainable. The other thing is that cellulosic crops can be grown on so-called marginal lands. So these are the lands that have been abandoned from previous agriculture but now not forested. So that can actually help to mitigate both food versus fuel conflict as well as the indirect land use change. So that's why we study cellulosic crops grown on marginal land. So that's the background. So if I specifically talk about the study that we recently published, so in this study we compared the productivity of 6 bioenergy cropping systems grown on 5 marginal land sites over a 7-year period with and without nitrogen addition. So in this study, what we measured was the biomass yield. So the reason why we wanted to study biomass yield is because if you take a look at the biomass to biofuel production pipeline, biomass is actually at the front end. So if there's any variation in the biomass yield, then that will have a direct influence on the biofuel production, because at the end of the day what matters is the biofuel production. And also we included a nitrogen treatment. That's mainly because so we studied 6 bioenergy cropping systems. 3 of them are monocultures, 3 others are polycultures, and it's somewhat common adding fertilizers to monoculture systems, but it's not that common with polyculture systems. So we wanted to narrow that knowledge gap as well. And we studied 5 marginal land sites that differ in climatic conditions as well as soil properties, and some marginal land sites experience extreme temperatures, and some marginal land sites experience water stress conditions. So we wanted to see how different bioenergy crops respond to different climatic and soil conditions. So that was another goal. And also we studied 6 bioenergy cropping systems that vary in diversity. There were less diverse systems as well as highly diverse systems. So overall, we wanted to provide a complete picture on the production potential of different cellulosic bioenergy crops grown on marginal lands. So we believe the findings of this study will definitely lay the foundation for future research as well as to take some management decisions. So that's why we studied this matter.
Yeah, I really appreciate this research. For one, because I don't know a lot about biofuels, so it's kind of fun to just learn like the different generations and things like that, which I had no idea. And also just I'm a big fan of research that is kind of like really efficient and kind of finds those gaps where you can just fill, get a little bit of extra yield here or find another use for land here. Mm-hmm. I love stuff like that. So you were looking at kind of these second generation crops and how they might be used on these marginal lands that wouldn't traditionally be farmed for other purposes. And some of the crops that you were looking at were like switchgrass and miscanthus and hybrid poplar. So I was kind of surprised to see poplar on the list because I think of that as a tree, and I didn't assume that you would be harvesting trees. So can you just tell me a little bit more about kind of what the different, I guess some of the different plants that you were using in these mixes, as well as kind of what they look like, how frequently they're harvested. Presumably you're not harvesting a tree every like 3 months or whatever. So can you tell me a little bit more about kind of just the process of growing and harvesting some of these crops and what that might look like?
Yeah, sure. So we actually had 6 bioenergy cropping systems along a historical vegetation. So as I mentioned before, there were 3 monocultures and 3 polycultures. The 3 monocultures are switchgrass, miscanthus, and hybrid poplar. So switchgrass is a warm season C4 grass, and it's native to United States. And in 1991, Oak Ridge National Laboratory's biofuel feedstock production program identified switchgrass as the model species for bioenergy crops. So that's why we study switchgrass, and it has a huge potential to be a cellulosic bioenergy crop because it has a high nutrient and water use efficiency, and also it can thrive in a wide geographical range because of those kind of characteristics. The second monoculture is miscanthus. So it's an introduced species, and the research related to miscanthus is relatively new in the United States, because if you look at the research done in the Europe, they have been studying miscanthus for almost 3 decades, and it's relatively new to the United States. And sometimes miscanthus can be invasive. So, and also since it's an introduced species, it has less biodiversity value compared to switchgrass. But historically, we know that miscanthus can give high biomass yields. The third monoculture is hybrid poplar, and hybrid poplar is the only woody perennial we used in this study. One of the reasons for studying hybrid poplar is that it can provide the supply chain flexibility when producing biofuels. And the 3 polycultures are a mixture of native grasses, which comprises of 5 native grass species. We also have a restored prairie and also early successional vegetation. The native grass mixture consists of 5 native grasses, namely switchgrass, big blue stem, little blue stem, Canada wild rye, and Indian grass. And the early successional vegetation is simply the species that emerge from the seed bed after moving down the existing vegetation, and also whatever the seed dispersed into that seed bed then emerged out of it. And the restored prairie is a combination of 6 grass species and 12 forb species. And so we studied these 6 cropping systems, and also we included one historical vegetation as a control. And so these data actually came from an experiment called Great Lakes Bioenergy Research Center's Marginal Land Experiment. So I'm representing the Great Lakes Bioenergy Research Center. It is one of the 4 bioenergy research centers in the United States funded by the Department of Energy. And so this Marginal Land Experiment was established in 2013. And during the establishment, I should say that when establishing these 7 cropping systems were established in a randomized complete block design with 4 applications. And during the establishment, each plot was divided into 2 to include a nitrogen treatment. And every year, except hybrid poplar, the other cropping systems, some part of the cropping systems receive a dose of nitrogen. And we first fertilized hybrid poplar in 2016, and except hybrid poplar, all other cropping systems are actually targeted to harvest within 2 weeks from the first killing frost in fall. That's mainly to reduce the nutrient removal as well as to improve the biomass yield. So these are perennials. So one of the characteristics of perennial vegetation is that when they getting closer to the end of the growing season, most of the nutrients available in the above ground part are transported back into the root system. So just to allow that process, we give like 2 weeks of time. And in that way, we can reduce the nutrient removal. And if we keep for a longer period of time, even after the first killing frost, then we will lose more and more biomass. So we have to, harvest within that 2 weeks period. And finally, the harvested biomass is then chopped and weighed, and then a subsample is taken, and then the subsample is dried and retake the weight, and using those data we calculate the biomass yield as megagrams per hectare, and that's what we call the biomass yield. So that's what we finally compare between the different cropping systems across different marginal land sites.
Hi everyone, I hope you're enjoying the show. Interested in learning more? Dileepa's paper, Comparative Productivity of 6 Bioenergy Cropping Systems on Marginal Lands in the Great Lakes Region, USA, published in Agronomy Journal, is always freely available. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Let's get back to the show. So you had these different tests. You were checking to see if nitrogen helped with the biomass, and just how these different mixes or not mixes might work in these marginal systems. So what did you find? What were your results?
Yeah, so we actually found some results that have never been reported before. So when you take the average of the years, the nitrogen treatments, and the marginal land sites, the overall biomass of the cropping systems decreased in the order miscanthus, switchgrass, hybrid poplar, native grasses, restored prairie, and the early successional vegetation. So in this study, although miscanthus had the highest biomass yield, the highest overall biomass yield, it was higher only in 3 of the 5 marginal land sites. So the biomass yield of miscanthus actually varied by a factor of 10 across different marginal land sites. However, in contrast, in switchgrass, the biomass yield was consistently higher. So when I average the biomass yields of miscanthus and switchgrass from Wisconsin and Michigan sites, the biomass yield of switchgrass was about 50% less than that of miscanthus in Michigan, and it was about 450% greater than the biomass yield of miscanthus in the Wisconsin site. So that shows how variable the biomass yield of miscanthus was. So the reason for this difference can be explained with climatic and soil properties that we observed in different marginal land sites. The main reason why miscanthus biomass yield was lower in Wisconsin sites was mainly because of the cold temperatures that we observed in the Wisconsin North, as well as lower snow cover that we received in the Wisconsin Central, and also due to the low moisture availability in the soil, in Wisconsin soil. So unlike other grasses, when planting miscanthus, we have to use the rhizome, and this rhizome is highly susceptible to lower temperatures. So during the establishment, because of that reason, during the establishment, the winter survival is highly critical, and if we have a better snow cover, then that can actually provide an insulation effect to the rhizome to overwinter. So if you look at the precipitation, the snow fall data from the 5 different marginal land sites, Wisconsin Central received the lowest snowfall, so that could be one of the reasons why we observed a higher miscanthus rhizome mortality. So that actually had some effects on the stand establishment. And also Wisconsin Central is the site that has the lowest soil water content out of the 5 sites tested, and miscanthus is also a grass that can get easily negatively affected by the water stress. So it could be either high water stress or low water stress. So in this case, it's the low water stress. So the kind of drought conditions that we observed in Wisconsin Central had a huge impact on the miscanthus establishment in that site. And Wisconsin North actually had the coldest winters out of the 5 sites tested, and that also had an effect on the rhizome mortality. So those are the main reasons why we saw a very low biomass yield in miscanthus in Wisconsin sites. However, switchgrass, it can tolerate wide stress conditions because it's a native species and it has evolved over thousands of years in the native habitat, and it has developed resistance against different biotic and abiotic stresses. So because of that reason, switchgrass was able to resist against many stressful conditions. If you look at the biomass yield of hybrid poplar, surprisingly, the highest biomass yield was observed in the least productive site, which is Wisconsin Central. That's mainly because of the lower biomass yields that we observed in other marginal land sites, mainly due to deer browsing as well as due to disease pressure. So if you look at the biomass yields of the other three polycultures, their biomass yield was mainly driven by the water availability. So usually the sites that receive the highest precipitation or sites that have the ability to retain more soil moisture tend to have higher biomass yields. So that's about the site variation. So if I talk a little bit about the variation over time, the two monocultures, switchgrass and miscanthus, they actually took about 3 to 5 growing seasons to reach the peak biomass yield, and then thereafter their biomass yield declined towards the year 2020. In contrast, the two polyculture systems, the native grass mix as well as the restored prairie, their biomass yield gradually increased over time. And interestingly, in 2020, the biomass yield of native grass mix outcompeted the biomass yield of switchgrass in all 5 marginal land sites, and even in the restored prairie, the biomass yield outcompeted the biomass yield of switchgrass in 2 of the 5 sites that we tested by the year 2020. We didn't see a growth improvement in the other 2 systems, which are the early successional vegetation and the historical vegetation. We didn't see a yield improvement over time. And if I talk about, so that's the temporal variation, or the variation that we observed over time. And if I talk about the nitrogen treatment, so nitrogen fertilization actually helped to increase the biomass yield significantly in all cropping systems at all marginal land sites. So those are the main findings of this study.
Yeah. Man, I just really enjoy this research. I should have asked this earlier. Is miscanthus like a grass? Is that what it looks like?
Yeah, it's a grass. Yeah.
Okay. Yeah, so obviously there's a lot of factors that are impacting the growth of these just in general, with some of those climatic and soil conditions, as well as just kind of over time they start to shift, which I think kind of leads us naturally into some of the future research issues that you might be able to look into with that. So can you tell us just a little bit about, what are some areas of ongoing growth in this research and continuing study?
Yeah, sure. So in this study, we presented the data generated between 2014 to 2020. So we are still collecting data, and we would like to study what has happened over the past 3 years as one of the future studies. So that's one thing to look at. The other thing would be to study the reasons for the yield declines, especially in monoculture systems. So I mentioned you earlier that the monoculture systems like miscanthus, switchgrass, they take about 4 to 5 growing seasons to reach the peak biomass, and then thereafter their biomass yield is actually declining. So we would like to know what are the reasons for this yield decline. So based on what we know, it seems like the nutrient limitation may play a role in this yield decline, as well as the disease pressure could have an impact on the yield decline. So we would like to further study those. And also, one of the things that we noticed during this study is the emergence of some of the species in the polyculture systems. So we have we studied 3 polyculture systems, and in 2 of those polyculture systems, namely the native grass mix as well as the restored prairie, we included another native grass, which is big blue stem. So we observed the emergence of and dominance of big blue stem in most of these polyculture systems. So we would like to study how these big blue stem perform if it is grown as a monoculture system. So those are the future research that we are interested in right now.
Yeah, for sure. Man, there is just so much going on with this research. I'm excited to see where it goes from here, but I know that I don't want to take your entire day, so I have 3 questions left for you. The first one is, if listeners want to learn more about any of the things that we've talked about today, where can they go?
Yeah, we have our website, which is www.glbrc.org. And if you visit our website, you can learn more about what we do in the Great Lakes Bioenergy Research Center. And there's a tab called Get Involved. And if you are a researcher or from industry or from press, all the contact information are given there. So if you're a researcher, the research contacts are our science director, Federica Brindisi, and also Science Director Robert Landick. And if you are from industry, you can contact our Associate Director Yijing Cheong. And if you are from press, you can simply contact our press contact, who is Matt Wisniewski, our Communications Director. So you can learn more about the kind of things we do in our research center, and also there are actually several startups that started based on the research that we do in our Great Lakes Bioenergy Research Center. So you can learn about them as well, and you can get information like the kind of people who are involved in these processes. So yeah, website is the place to go.
Awesome, for sure. And then obviously there is that Get Involved tab, but what else can people do if they want to get involved, either as like a researcher or a farmer who's interested in these kinds of things? What can people do?
Yeah, so you can definitely get involved in the kind of research that we do. And there are some outreach programs, and I'm pretty sure there are there's a contact person for outreach programs. If you're a farmer, you can definitely contact the person who's involved in the outreach. Yeah.
Awesome. Yeah, we'll include links to those resources in our show notes to make that a little bit easier for you. And then final question is, what is one fun fact that listeners wouldn't know about you if all they had was your research?
I played field hockey for 13 years, and I was actually fortunate to represent so I'm originally from Sri Lanka, and I was fortunate to participate the first South Asian University National Games. So I was selected to the Sri Lanka National University team in 2007 or 2008. Can't remember exactly. So, so that was a proud moment for me.
That's amazing. That's so cool. I love that. What a very fun fact. Well, this has been a wonderful conversation. Thank you for your time today. Thank you for the research that you're doing. And yeah, I wish you the best on all of those fun future research topics.
Thank you so much. It was a great pleasure for me to talking with you. Thank you, Abby.
Hi, everyone, and welcome to our Student Spotlight, where we highlight the work of graduate and undergraduate society student members. Today, we'll be talking to Anthony. Welcome to the show. Can you start us off by introducing yourself and where you're studying?
Hi, thank you for having me. My name is Anthony Mirabito, and I first began my journey in soil science working on my bachelor's, where I studied biology and environmental science at Iowa State University. I'm currently a PhD candidate in the Aquatic Biogeochemistry Lab under Dr. Lisa Chambers, working on my PhD degree in Integrative and Conservation Biology at the University of Central Florida in Orlando, Florida.
Awesome. And what are you currently researching?
For my current research, I am working with the Army Corps of Engineers, focusing on understanding how soil organic carbon pools vary between restored and natural coastal wetlands in terms of both quantity and quality. Specifically, I'm investigating how stable pools of organic carbon, such as mineral-associated organic carbon, compares in coastal wetlands that have been restored using beneficial use dredge sediment to natural reference wetlands from coastal wetlands ranging from Chesapeake Bay to San Francisco Bay. Mineral-associated organic matter is considered better protected from microbial decomposition, which can allow more carbon to be stored in the soil instead of being in the atmosphere.
Great. And if you could have your dream research project, what would that be?
At the moment, my dream project would continue exploring how prevalent mineral-associated organic matter is in coastal wetlands and how this varies spatially. Since geology and mineralogy can vary, it would be interesting to better understand how different minerals or regions may impact these stable carbon pools. Also expanding to have more data coming from coastal wetlands from other countries where sampling and data may have been limited. This would allow scientists to get a more accurate global inventory of stable carbon pools and what implications this may have in a changing environmental conditions.
Awesome. If you'd like to get in touch with Anthony about his work, we'll have his contact information in our show notes. Thank you so much for being on the show today, and best wishes on your future studies and career. Thank you.
Yep. Thank you for having me.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.